Okay, so I've been to Smash Burger, got something to eat, ready to go again. A lecture on immunology and the immune system. So, um, as we ended the last lecture, we're looking at antibody-mediated immunity. This was I sort of put this figure together as to bring everything together so that you can see the various pieces. Um, as we talked about before, macrophages, the B cells, where the different types of T cells fit in, and how they lead to eventually memory cells, T cell memory cells, T cell activated T cells, activated B cells, plasmacytes, memory B cells, and then ultimately antibody. So this is the guts, and if this is a way of just looking at everything as a piece, and you can see uh, common things about this are antigen presentation between the macrophage and the B cells. And you can see here that there's a very lot of similarity between those in the T cell receptor areas, and then also a bit of differences and that you have CD40 ligand for the B cell, which you don't have for the macrophage or dendritic cell. But I want to make sure you understand that these cells here are and these are referred to as antigen processing cells or antigen presenting cells. And so when we talk about that, or when you hear that, you're talking about macrophages, dendritic cells, B cells. All right, not T cells. T cells are not antigen presenting cells. Um, and then so you can tell, these are very common, and then the amplification. The system gets amplified, and so it increasingly. You get a lot more cells, B cells and T cells, that are produced this antibody. So what I'd like you to do is, in this time, we're going to start with the review questions to start with. See what you remember from the last lecture that I gave. And so why don't you stop here? You can stop and answer these questions and see if you can get them right. And so you can stop and do that. The next slide will be the answers to those. Now, when we looked at this organization of the receptors on the surface of these T cells, I pointed out, I made a point of saying that and these are organized. And although what we looked at is just one, it's not just one receptor that interacts, but many of these receptors that interact on the surface of the cell. And so what you'll see is you'll see what you'll see is that there'll be a clustering effect. And if we were to look at the interactions between the two cells, there's a thing referred to as the immune synapse is formed. And what that means is if we look up here, here's the interactions between the two cells. If we could look right in and we labeled the various components, what we would see is that those adhesion molecules are all around the top. So in this case, um, uh, what we're looking at is we're looking into um, make sure I got this right. We're looking into the T cell. And so you can see the LFA1 is here. There's LFA1 here. And then inside you can see these have been stained, so these are the T cell receptors. And so the ICAM1 is on the B cell with MHC2, and then the LFA1 is the other adhesion molecule. The adhesion molecule on the T cell receptor is on the T cell, and they basically corral these MHC2 T cell receptor interactions. They form sort of this bullseye at the interaction between the two cells. And in the center, these are sort of a description of here. In the very center, you have the T cell receptor CD28 a couple other ligands. And then on the outer part, you have LFA1 and ICAM. And there's another molecule, CD45, which we won't talk about. But so this is when you're looking at it, we were able to sort of peel it away and look right at that interface. They interact with one another. Just if you took like two balloons and pushed them together, those two balloons would form an interface between each other, and um, that's the organization of these receptors in that interface. So it's a very distinct interaction between those, and it's very well organized between them to maximize the interactions between the T cell receptors and to put some stability there to see if there's an interaction that's going to lead to activation of the cells. This is just another, this is now looking at T cell where you at an electron micrograph. And the red here, the red is the LFA1, and then the T cell receptor CD28 are all in this corralled in this little circle here. So this is like the fence, LFA1 fence, um, that corrals then the T cell receptor. And this is referred to again as the immune synapse, just like when you have in neuroscience, you have the neurological synapse interaction between two neurons. In this case, this is a T cell interacting with a macrophage, dendritic cell, or a B cell. So it's highly organized. So we're going to look now at what happens inside the cell. What are the signal transduction pathways that occur, and how does this transcription of those interleukins and those signaling molecules occur? What I've given here right here is a little glossary of terms in which it's a little glossary in which you have a variety of different sort of these acronyms, and these are the what they stand for. So PLC equals stands for phospholipase C, DAG stands for diacylglycerol, PKC stands for protein kinase C, etc., etc. And some of these have very distinctly important. For instance, there's several. Then there are these three transcription factors that are involved in IL-2 production, regulation of IL-2, as well as other cytokines that we've seen. And then there are molecules that are inhibitors of the transcription factor that can stop it from occurring. So these are the ones. So in the next couple slides, we're going to go through this mechanism as a signal transduction pathway. I assume you've seen a signal transduction pathway before. I mentioned it just in passing early on in the overview that these are the processes that occur in cells, and they're governed by phosphorylation, by and large by recruitment of calcium, phosphorylation or dephosphorylation events, as in most signal transduction pathways. So if we were to look at now to look at the interaction again, here we have CD4, T cell receptor, peptide. We have this little dumbbell is the peptide, MHC2, and that's the T cell receptor. And the T cell receptor actually contains a few other proteins. It's a heteromultimer, and then these proteins are involved in. These proteins are actually part of the CD3 complex. It's part of CD3. One of those is the zeta chain, and so these are then components of that. And so this will show you how CD3 governs this activation. So if we have here's a T helper, TH cell. Here's the membrane. Here's the nuclear membrane, and down here is the DNA for the chromosome. So upon this, actually LCK, which is a kinase, usually is attached to CD4, And when it's brought into this complex, into the formation of this protein complex, it will undergo. It starts to undergo. Starts to hydrolyze ATP and phosphorylate that zeta chain. So the zeta chain gets phosphorylated for CD3. This recruits ZAP70 to the zeta chain, and ZAP70 then is also a kinase, and what it does is it phosphorylates phospholipase C, PLC, and so there's been two phosphorylations here, and so then phospholipase C, what it does is it's going to, when it gets phosphorylated, it does two reaction. It does a reaction in which it takes inositol phosphatidyl um, inositol phospholipid, and what it does is it splits it into two, in which it takes alpha in which this is the DAG is diacylglycerol, which is right up here, and then it takes basically the head group off, which is for the peptide, which is inositol phosphate, and that's IP3. That's IP3. It splits that off, triphosphate, inositol triphosphate, splits it off, and then the IP3 is going to do inositol triphosphate, has the ability to activate release of intracellular stores of calcium, calcium 2. That is going to interact with a molecule that likes to bind calcium. It's called calmodulin, and so it's going to bind. You're going to see an increase of calcium binding, calcium bound calmodulin, and that increase in the binding of calcium to calmodulin recruits a molecule called, it's referred to as calcineurin. Now this calcineurin is a really important molecule, because what it does is it doesn't do a phosphorylation. What it does is it actually dephosphorylates, and so it's going to act on the nuclear factor AT, NFAT, and remove this phosphate, and remove the phosphate, okay, to produce to give you NFAT. Now NFAT will, once it's dephosphorylated, it can now move across the nuclear membrane, and it will eventually end up bound to the chromosomal DNA in a promoter region for the genes that it regulates. And it will recruit another protein called AP1 to that area, and they'll form this mixed multimeric complex, which is a transcription factor. They're transcription factors, so they lead to the transcription of particularly the cytokines of the proteins that they've attached to. The protein encoding sites that they've attached to. Now, what happens to the DAG? So the diacylglycerol, what it does is it binds to the protein kinase C, and when it does that, it activates protein kinase C, and protein kinase C is going to do an interesting thing. It's going to lead to phosphorylation of I-kappa B. Now, I-kappa B is an inhibitor of NF-kappa B, and so when NF-kappa B is bound to I-kappa B, it cannot get across the nuclear membrane. But once protein kinase C phosphorylates the inhibitor in I-kappa B, it will then dissociate. It then dissociates from NF-kappa B, this transcription factor. So it breaks them apart. So in this case, the phosphorylation events releases then the transcription factor. In this case, the NFAT case, the dephosphorylation of the transcription factor directly leads to nuclear localization. And so the NF-kappa B is now going to be able to get to across the membrane of the nuclear membrane into the nucleus, and it's going to bind to the DNA, to the chromosome, and end up leading to IL-2 production and those cytokines that we've already talked about. So what you see here is that there's basically a activation through the T cell receptor. So there's activation up here. There's activation up here. So you could see step 1. Step 2 is the phosphorylation, the events leading to phosphorylation of the zeta chain. Step 3 is then the splitting off of DAG and IP3. Step 4 is mobilization of calcium and activation of calcineurin by binding to calmodulin. And step 5 then is activation of NFAT, so that now it can go into the nucleus and bind to the DNA. Step 6 is protein kinase C, then inactivating the inhibitor by phosphorylation. And then step 7 is the NF-kappa B then moving into the nucleus and leading to transcription. And so those are the really important steps that you want to pay attention to, and you really want to be aware that these signaling processes are complex, but there's a fairly straightforward way of understanding them in terms of what they do. And that what they do is they go through the series of events that lead to either phosphorylation, dephosphorylation, and the initiation then of transcription initiated translation, transcription, initiation of transcription. So that's what's going on this in the cell, and how you get those the production of those interleukins. So here's a bit of review. Here's a review question. 3 questions. You can stop here and take a break, get something to drink, and then answer the questions. The next slide will have the next question on it, and we'll stop this segment right here and finish up with. Actually, I'm not going to do that. I think I'm going to. Yeah, I think what I'm going to do is I'm going to stop here and we'll continue on the next online video.